Welcome to the 2024 Regional Advanced Genomic Epidemiology Training. This is produced by the Virginia Department of Health and the Division of Consolidated Laboratory Services, or DCLS, in Virginia, the State Public Health Lab. This will be a series of 4 lessons, including genomic applications in public health, pathogen diversity, laboratory and bioinformatic workflows, and inferring genomic relatedness. All 5 of these learning objectives span across all 4 lessons. So for the first lesson, my name is Jenny Crain. I am a genomic epidemiologist at VDH. I just want to set the stage by talking about the World Health Organization Genomic Surveillance Strategy document. And in this document, they include a mission statement that is to provide quality molecular epidemiology intelligence for use by country, regional, and global stakeholders. And I like how in this document they use the word intelligence, not surveillance. Often when the public hears the word surveillance, they think police or FBI, collecting personal, private information about them. As intelligence, we are collecting information, but we are using this information in conjunction with many different data sources, including epidemiology interviews and things like that, to gather information or intelligence to, solve public health outbreaks or track diseases that are of public health importance. So I just like how in this document they use the word intelligence instead of surveillance. But in fact, we have been doing genomic surveillance of SARS-CoV-2 for quite a long time. So these 2 slides are focused on that specific virus. And I just wanted to stop at the beginning to say that while the pandemic raged on for 4 years and we are now in the post-pandemic period, we are still doing a lot of genomic sequencing of this specific virus. And a few years ago, while this was a very local experience, we were all in practicing social distancing. We may not have been in our offices, and a lot of the case investigation and contact tracing were all occurring via telephone or, from remote locations. We were really experiencing this at a local level. And while we were still observing and paying attention to the global scale as to what was going on, but now that, we are not in that situation anymore post-pandemic where we are all out and about and circulating in the community again and interacting somewhat normally again. We are still at a global scale, still sequencing SARS-CoV-2 and paying attention to this virus as it mutates and changes. And at the time of this recording in late August of 2024, we are seeing an increase of SARS-CoV-2 again, and there are a lot of outbreaks that we are investigating at a local level. So just to set the context, I wanted to talk a little bit about this strategy document. This next slide I borrowed from the CDC Office of Advanced Molecular Detection, and I like, while there's a lot of things on this slide, I like how it really does give an overview of many different applications of sequencing. And if you focus on the bottom left-hand corner where this red circle is, this really highlights the primary focus for state public health labs up until the SARS-CoV-2 pandemic. The yellow star here now focuses your eyes on where state public health labs are now building capacity to do more of this. So in the state of Virginia, we are working on increasing our ability to do bacterial pathogen typing of some of these pathogens that are listed here. And again, back to this WHO genomic surveillance strategy, I like how this specific diagram or figure from that document highlights how, while we made great strides and advances during the SARS-CoV-2 pandemic in terms of sequencing, now this idea of trying to use those advances to expand our ability to sequence other different pathogens will in fact prepare us for the next pandemic. And so, if I were able to poll the audience and say, well, do you think the next pandemic is going to be caused by a bacterial pathogen that primarily affects elderly in long-term care facilities? Well, the answer might be no. You might be more likely to say, well, it'll likely be another respiratory pathogen, perhaps novel or not, but that affects a large number of people and that can spread rapidly, across the globe, for example. However, we are still very interested in those bacterial pathogens and those smaller scale outbreaks as well. So this concept of, cross-cutting actions to strengthen genomics, and this idea of having, vertical actions across different different pathogens, as well as different hosts or exposures in terms of zoonoses or food, etc. This is how this concept will prepare us in terms of the next pandemic. Okay, so it wouldn't be a real training without learning objectives and definitions. So I just want to start here briefly by kind of ensuring that everyone understands the difference between NGS and WGS. And so I think these terms are often used very interchangeably by scientists. And while that's all good when they know exactly what they're talking about, sometimes it's a little confusing for epidemiologists. So I just want to say that, whole genome sequencing, whenever you see the term WGS, this does denote when a full genome approach has been used to study a specific bacteria, virus, pathogen, etc. And so anything else that could be referred as sequencing or next generation sequencing, this is a larger umbrella term, is kind of how I think of it. And this is, any variant thereof, whether using primers or, targeted sequencing just to identify and sequence a specific part of the genome. And that they're not actually sequencing the entire genome. And so that just comes important when you're doing multi-sequence alignments and trying to understand and start to interpret or infer things, make conclusions from those genomic results. And the last thing I just want to highlight on this slide is the last plain language definition of sequencing, the last bullet down there. And this is just in Virginia, we had a request from our regionals to, help translate sort of what sequencing is, so that when they are conducting interviews or investigations and they're trying to explain to the common person, they wanted some a layman description. So you can read here, sequencing is a specialized lab test used to examine the genomic fingerprint of a germ causing a person's disease, and then to compare it with germs from others who have similar illnesses. So I included this in one of our guidance documents that we created, and we thought this would be helpful to share. Okay, another term that I just want to, spell out or clarify. In Virginia, I've had a lot of success trying to when working with multiple different stakeholders, say IT from the state lab side or the Department of Health side, trying to get everybody to speak the same language is not always an easy thing. And so we've really tried to express, that the term sequencing data really refers to just the ATCs and Gs, the sequential nucleotide information that is generated through whole genome or genomic sequencing, and that is the information that is coming off of the MySeq, Illumina, sequencers in the state laboratory. And so IT always wants to know, well, how big is this data? Where do you want to store it? What is the, data frame? What are the variables involved? How big is it? And we have had a long it was hard for us to express that we really don't want a copy of all of that sequencing data at the public health department. We really just need to have the genomic results. As epidemiologists, we're interested more in, the laboratory report with that applied genomic epi interpretation of all of that genomic testing. So that includes that wet lab sequencing as well as the dry lab bioinformatic analysis, and then interpretation of all of that sequencing information. So, well, this may be clear to everyone who is listening to this presentation. It's not always clear to everyone that you're working with. So we found great success in trying to just separate these two concepts and make sure that all people involved understand the difference. So a few more definitions, and I'm not going to read all of these, but they're listed here in a sequential order intentionally. So many epidemiologists, are familiar with epi-surveillance. They've conducted case interviews and filled out case report forms. Case surveillance is quite obvious as well, but genomic cluster investigation, when you get to that term, this is where it really gets into the meat of what we are attempting to do as genomic epidemiologists. And this is, investigating these potential linkages that could exist between cases that have been identified through genomic results. And based on those genomic results, we have found some sort of genetic similarity, but we need to investigate then to determine if there are in fact any epidemiologic linkages between them. And then the final term here, integrated genomic episurveillance. This is really the end game, or ultimate goal of what we're trying to achieve here. And we haven't quite succeeded in doing this for all pathogens yet, right? A lot of our integrated genomic data, when we're integrating it with epimetadata, it often is very manual, and we cannot do this for everything. But this is really the ultimate goal, where we're trying to integrate all of that information together in order to complement those genomic results and to better investigate and understand disease transmission. And the last link on this slide, just points to an excellent reference for other acronyms and terminology that was produced by the New York Food Safety Center of Excellence. And I just would point you to this if you find any other terms that are not familiar with you to you during these series. All right, so the section goals for this is really just to try to understand the reasons for testing, and knowing that we need to understand that in terms before we start applying the genomic results from sequencing. And sequencing can be used for phenotypic inference, for diagnostics, for phylogeny determination. We'll talk a lot about phylogeny determination in the fourth lesson. And ultimately, this lesson, we're going to just really try to make the distinction between doing routine, ongoing surveillance versus non-routine outbreak or cluster investigation. And those are the two main applications of genomic results that we'll talk about today in terms of public health. We can also, we're going to talk about the potential sources of specimen acquisition and some of the difficulties with gaining those specimens to do sequencing. For example, culture-independent diagnostic testing, and how this limits, some of our ability to get that raw sample, original sample for testing. We'll talk a little bit about public health surveillance systems that already use genomic results, and then we'll talk about the value of integrating all of these multiple different data streams together. All right, so why is analyzing public health or pathogen genomic data useful for public health? This is a little bit of a no-brainer slide, but it is important to address the fact that pathogens evolve. So we will have a lesson about pathogen diversity, specifically talking about how mutations occur and accumulate in pathogens over time, especially when transmitted between people or animals. That lesson will also talk about evolutionary rates, how to calculate them, how those really differ based on the pathogen, and how that is important for understanding transmission. And then this last slide point here just is a shout out to Allie Black, who works for the Washington Department of Health. She created this really great genomic epi handbook during the pandemic, and I found it particularly helpful. So thank you for her permission to reuse some of her slides or figures in these lessons in the series. So what can genomic data tell us? It provides greater resolution than other subtyping methods, and it can help us with this phylogeny determination, right? Are the cases genetically related or not? It can also help illuminate potential transmission pathways after we've integrated that with other epimetadata. However, a phylogenetic tree is not the same as a transmission diagram. So we will talk about that in a later lesson, how, we can use it to make inferences, but a phylogenetic tree cannot, be used in standalone. We'll also talk about how genomics results can help identify potential sources of exposure, whether that be environmental or food. And then genomic data can also, provide context, especially if we're doing ongoing surveillance, and help us understand, what variants are circulating in a community of a particular pathogen. And then finally, genomic data can help us understand the pathogen more specifically. So characteristics such as antibiotic resistance, as well as virulence genes. So this is taken from Allie Black's Genomic Epi Handbook, and I really like a couple of the visualizations that just drive home this concept of, well, we can study disease incidence over time on this XY scale. The top gray diagram here, shows, and you can see that there's been an increase in cases over time, and then it's waned, right? But the bottom, once we add on that additional genomic results and the genotype specific information, this provides a greater level of detail in terms of understanding when different genotypes, appeared or started circulating over time. And then this is just another way to think about that in terms of applying genomic results to, say, congregate facilities and thinking about investigating disease transmission among those. So the top shows, the cases without that genotype information. So at this point, we might know, the number of cases, total number of cases in outbreaks at, 3 different skilled nursing facilities. But once we have the genomic results, then that really provides a different level of understanding in terms of now we can say, all right, we see genotype A, in 3 different skilled nursing facilities. What other epimetadata might we add to this to further elucidate, disease transmission? Do we know whether, there were staff or residents that were positive in, all 3 of these facilities? Was it potentially staff, that were shared between facilities, or do we know that there were specific patient movements and transfers that occurred between these facilities? So again, I just like these 2 as a, sort of starting point in terms of how do we visualize and try to understand how to use genomic results. But with all good things, there are limitations. It is very pathogen dependent. And one of the things I like to say about, public health investigations, it really is very dependent on how well, the pathogen in terms of your success for that investigation. So do you know, the disease, symptoms initially, right? When you don't have lab results, for example, do you know the symptoms? Do you know disease onset times for those patients? Do you know, exposure sources or foods that they ate or where they were? Do they have any shared common exposure locations or places they were, for example? And so as you try to investigate that outbreak, once you have laboratory data and you know which pathogen it actually is, then you will have a little bit more information in terms of, well, I know the incubation period for the specific pathogen, and how can I use that information to better understand, based on onset times, how these cases were all related together. So, same thing goes for genomic results. It's very dependent on how well you know the specific pathogen in terms of interpreting the genomic results that you have in front of you. So can you, do you know that the pathogen, has certain virulence genes, or that it is often susceptible or picks up plasmids easily that carry antibiotic resistance genes? Like, how much can you take away from the genomic results, based on how well you know that specific pathogen? Do you know if it's one that is often associated with healthcare associated infections, for example? And so, there are limitations with using those genomic results depending on what public repositories are available. Do we have a lot of comparative contextual sequences available to us as well for that pathogen? So there's a lot of things that could be said in terms of how we're limited by which pathogen it is in terms of using those genomic results and making inferences or interpreting the data. And then we've already mentioned sort of that we cannot determine the direction of transmission, or we cannot infer directly how disease is transmitted based on genomic results alone. But we can integrate that information with other epidemiologic data to better try to elucidate or have, draw some inferences about what's going on with disease transmission. Another limitation is how quickly we get these genomic results, right? Turnaround time is a big deal, always a hot button for any laboratory. And, epidemiologists will sometimes push back and say, well, why do we really need these genomic results if we've already made our infection control recommendations and we've already, determined the total number of cases in this outbreak? How is this really helpful to me? And I just always try to impress upon the fact that all recommendations can be changed or modified. New information we get all the time. If we find out another new case in a different, long-term care facility unit or wing after we've already made infection control recommendations on one, that's obviously going to change when we find new cases. Well, same thing when you find out which isolates from which patients are actually genetically similar, or is there a patient that is infected with an isolate that is dissimilar, that might have, they might have acquired their infection from a different facility or from outside in the community. And so again, it may be delayed, but that information will still help you in terms of the investigation. but it is a limiting factor. So same for sample acquisition. Do we have enough samples to sequence and to compare to each other? And then, how have we selected those samples in terms of doing the sequencing? Is it a representative sample of, say, the entire state, or do we only have samples from, the most acute cases that have been hospitalized, for example? Does that really tell us about statewide disease transmission? Again, it all depends on sort of what questions you're asking and what the goals are. But those are all, potential limitations. So in terms of the two main applications of genomic results to public health, I said in the beginning, we're going to focus on surveillance, more routine or ongoing operation, and that is going to allow us to look at several of these different factors here, as well as cluster detection or outbreak investigation. And this, will be basically a different application, but we'll go through these today. All right, this is one of my favorite diagrams, and I have borrowed this from a CSTE AMD webinar series. And so you can check out the journal article that I have referenced at the bottom. This is from Halpin et al. And if you want to know more, you like this diagram or how it summarizes the relevance and utility of genomic results. And it essentially varies whether we're investigating from the global level down to the local, that would be the top-down approach, or from the bottom-up approach, from the local level or single case up. And so just to illustrate this a little better, I think of the top-down approach being, say, maybe a national multi-state outbreak of an enteric pathogen that is associated with a certain food exposure, for example. And so there may be many cases that have been identified throughout a lot of the states, say 30-40% of the states, and we've identified that one specific food item is associated with that outbreak, but it's not like we can immediately limit. It's already a very large multi-state outbreak that we have identified. But by removing that food item from circulation through a recall, et cetera, we can then limit the additional number of new cases that or exposures that occur, and then try to limit how that multi-state outbreak grows. So that would be the top-down approach. And again, we would have identified most of those cases through ongoing routine surveillance, for example. The bottom-up approach would be, really the flip reverse of that, where we have maybe one single case that has been identified in a healthcare associated infection environment, right? A hospital or a long-term care facility. And in that case, maybe we've then identified a second case. And so it's a very small cluster, but the scale is different, and how we apply the results are going to be very different as a result. But we are now trying to actually contain the disease. We are immediately trying to recommend infection control strategies and try to prevent the outbreak from growing or spreading to multiple different units or wards in that hospital or long-term care facility, or even to try to prevent it from spreading, between facilities. And so this is the bottom-up approach, right? So it's really different, and it's based on more like cluster identification or outbreak investigation. And then you can see on this diagram, the x-axis also shows the laboratory method for, in terms of the most useful information that is going to be most relevant to that bottom-up approach or top-down approach. But whole genome sequencing, across the board is useful for both approaches, top-down or bottom-up. But some of our other, subtyping methods that we have used in the past are, more suited for one or the other. So I really like this diagram. It just helps kind of put it all into context in terms of, there are different applications, and they, in different settings and different contexts, we do need to think about how we apply or interpret this information differently. Okay, so as I mentioned in the beginning, and I'm not going to go through everything on this slide, there are many surveillance programs that are already using genomic results. And so as we have started off this lesson talking about the SARS-CoV-2 pandemic, I do think that many of these surveillance systems are already familiar to many genomic epis or those that may be listening to these lessons. So I'm going to give some screenshots and cover a couple of those in a little bit. But I did want to just stop here to mention PulseNet and how that's been around for going on 30 years. I think about 28 years. PulseNet has been used for, connecting multiple state public health laboratories who are doing sequencing of enteric bacteria in terms of ongoing surveillance. And then epidemiologists also submit epimetadata to CDC about those cases for the isolates of enteric bacteria, and all of that information is collected and integrated. And so this has been going on for quite some time. In addition, norovirus, another enteric pathogen, there's another surveillance system called CaliciNet. And so, if you've heard about norovirus G2 or a specific genotype of norovirus when doing an epi investigation, that is already based on genomic results. And so it's wonderful how CaliciNet is integrated with NORS, the National Outbreak Reporting System that epidemiologists use. And so those two systems speak to each other. And it's, again, these are systems that already use genomic results that have been around for a while. Other ones, the National Antimicrobial Resistance Monitoring System, this has been around since 1996. And what's important to just mention here in terms of doing ongoing surveillance of antibiotic resistant bacteria is that the system surveils not only clinical isolates or humans, but also retail meats and food animals. So this is a multi-agency surveillance system. So CDC, FDA, and USDA are involved in that surveillance. And also our ARLN, or Antimicrobial Resistance Laboratory Network, that has been around since 2016. Again, ongoing genomics surveillance systems that involve genomic results. And so those laboratories do genomic sequencing. For Virginia, our ARLN lab is based in Maryland. And then just another thing to mention in terms of ongoing surveillance systems, the ABC core surveillance. This is, again, an ongoing system, has been around since 1995. And some of the pathogens that are listed here are the ones that are surveilled for ABC core. And that started with about 4 states, and it currently operates among 10 different EIP sites. So emerging infection program sites across the US. And so again, right now there's 5 pathogens that are surveilled as a part of that. But, we have found this a really useful surveillance source when working on some of our infectious group A strep outbreaks and investigations. Okay, and then the last here, just to mention, there are other pathogens and surveillance systems in the state of Virginia. Our Division of Clinical Epi and Division of Disease Protection, do use TB genomic results as well as hepatitis C and HIV genomic results as a part of their cluster detection and response. Okay, so again, for a lot of the SARS-CoV-2 surveillance systems, many of those will already be familiar to you, but I did just want to mention here in terms of NS3, that may be a term that isn't super familiar to everyone, but the S's just refer to the SARS-CoV-2 strain surveillance. And in Virginia, we have what's called the VAS3 laboratory network, and that is a network of Virginia outside academic partners that do sequencing of SARS-CoV-2, and we created that network during the pandemic. And so just in terms of thinking about sample acquisition sources, like where do we obtain these samples for sequencing, specifically for SARS-CoV-2 here, we had many different sources during the pandemic. And so without going through everything on the slide, I just want to mention that, as the advent of at-home testing came about, we started to see a significant reduction in the number of samples that were forwarded to our state lab for sequencing. And, it continued to change throughout the 4 years of the pandemic. But we were also already struggling with, say, getting influenza specimens, before the pandemic started. And so the Sentinel surveillance program that we maintain in the state, is designed to attempt to increase those number of specimens. And so there is a large effort to continue to try to obtain samples. And so, this slide specifically pertains to SARS-CoV-2, but we have also, already, had a challenge with that with influenza. But again, what's important here is just to mention some things about sampling and, some of the bias that can be created as a part of our sample selection. So, during the pandemic, there this idea of baseline tagging and or how samples sequences from samples are identified as being outbreak specific associated specimens, or are these specimens that are coming from hospital or specific acutely ill patients. These are tagged and noted in a different way when they're uploaded to public repositories. So again, sampling is something that we continually strive to try to get a more representative sample across our whole state. So are there other sources of samples, that are available for sequencing? One thing to just note here that, the Code of Virginia, our Virginia regs, is really what guides, which specimens are forwarded by law or required to be forwarded to our state public health lab. And so enteric pathogens definitely are on that list. And in 2018 in Virginia, carbapenemase producing organisms were also added to our reportable disease list. And now the outside laboratories and hospitals are required to forward those to our state lab for sequencing. Another issue here, as we mentioned in the beginning, is this concept of culture-independent diagnostic testing. So this often more rapid tests, prevent the original sample from being available to be forwarded to the state lab. And so reduced clinical testing at public health labs. This is also something that is affecting the advent of different respiratory diagnostic tests during the pandemic has also sort of changed our ability to obtain respiratory samples or influenza samples post-pandemic as well. And that is something that, we really strive to try to get a better sense of which outside laboratories are doing what respiratory tests at this point. We've conducted a state survey of outside laboratories in Virginia to try to get a better understanding of that and to try to partner with new non-Sentinel labs to obtain specimens for sequencing in the state of Virginia. And then lastly, I just wanted to mention here that, CDC still does have contracts with certain clinical labs for obtaining respiratory pathogens and to do sequencing. And again, this tagging for routine surveillance was important and really critical for understanding, contextual data in terms of looking at what sequences, why we were sequencing certain isolates. Okay, I did just want to, again, like I said, these should be familiar to you, but I wanted to include a couple screenshots from the CDC COVID data tracker just to ensure that we're all on the same page. So the clinical genomic surveillance that has appeared on these websites for the last 4 years. I know we are all readily familiar with looking at these over and over again, but I just want to mention that the nowcast estimates that CDC is able to calculate there, you can select the different HHS region, which is really lovely. And as the number of samples that we had on a even HHS region level dropped off, those nowcasts would become unavailable if there was less than, I think it was 300 samples. And so, again, we need to have a representative enough sample that we're collecting from to have that contextual information and to make some of these calculations so that we can predict and make these nowcast sort of modeling estimates. And so we've never been able to generate any sort of a nowcast in the state of Virginia, but we do look at our data, even if it is 2 weeks times late with the genomic results, we do look at our variant data and compare it to what we see as the national level as well as on the regional level. And we're still doing that. I just want to mention this other genomic surveillance system that CDC conducts that is involving travelers, because not everyone was super familiar with this throughout the pandemic. And I don't remember exactly when it got started, but it is currently still in operation at 5 different international airports across the country, one of which is Dulles here in Virginia. And so that information again, is anonymous. These are all volunteers, and they are basically sampling from travelers that are returning from international locations. And so the information that they give in this voluntary survey, is not super complete. It's sometimes a bit of a struggle for an epi mind, as we want to know exactly who it is. Are they a Virginia resident? Is this their final, traveling destination? Or is this just a, transfer for them as they're, moving on to another state and they actually live somewhere else? And we want their first name, last name, date of birth, but that's not the way this system works. And so this is, again, an anonymous sort of voluntary surveillance system, but it is really important. The first, I think it was BA2 2.86 was identified, through the traveler genomic surveillance system in terms of when it first was identified in the U.S. And so even if we don't have granular, super detailed information, it is an excellent surveillance system. And so the point here is just kind of to say that we are, again, taking all of this intelligence from multiple different systems and we're piecing it together. So wastewater surveillance is another example of that, where it has evolved a lot over the pandemic, and there's still a little bit of, varying opinion in terms of how useful it is, in terms of what we can pull away from it and what recommendations can we make, in terms of public health action. But it is another data source, and it is very important, and it doesn't lie, and we shouldn't ignore it. Every state, I feel like, is at a different level in terms of where they are with using this information, but they are able to test, way more than just SARS-CoV-2 and polio at this point. Also mpox, but also now influenza, even H5. And and RSV, and they continued to add additional targets for wastewater sequencing, to include even opioids and other drug substances as well. So again, this slide just sort of shows benefits versus challenges, pros and cons, if you will. But again, I won't make state an opinion either way, but it really depends on, just like all genomic results, what do we do with that information? How do we apply it? How do we translate it to public health action? I know in Virginia we had used it for, identifying which wastewater treatment plants were associated with certain communities where there were low vaccination rates. So if there was, consistent and positive detects of SARS-CoV-2 in a certain area, then we could deploy, a vaccination action team and try to, offer and do an advanced targeted vaccine campaign for that specific community, for example. But again, this is dependent on, how cooperative is the public with taking those vaccinations. And so, in the end, did it really help us to reduce the number of cases associated in that community? I don't know. But it certainly is another piece of information that we can add to our intelligence and our investigative public health responses. And so again, multiple different targets have been added, and it continues to change and advance over time in terms of standardizing methods and trying to understand how to apply this information. Okay, this is just another screen grab from news or the National Wastewater Surveillance System, and just shows you sort of what that aggregate data looks like, and you're able to zoom in on your specific state or county or region, and then look at that in a more granular level. This is a slide that was contains some information that was put together by our Office of Environmental Health. And we have a Virginia wastewater monitoring program that is still very active in terms of working with outside wastewater treatment plants, outside in terms of not state-run, but publicly funded and run wastewater treatment plants. And so all of those partners are still actively participating in forwarding wastewater samples to our state public health lab for sequencing. And so our state lab supports this. And these are, just some visualizations that Carolyn Marty from OEHS have put together. This is pretty dated at this point, but it was nice, a nice correlation where we were able to look at the proportional abundances of all of these variants in wastewater and look at our case data in terms of viral load over time. Okay, and so just going back to, that was a long chat about surveillance. The other main application of genomic results is cluster detection or outbreak investigations. And so Well, this is, the application that is a little bit more common or familiar to everyone in the fact that PulseNet has been around for about 30 years now. This genomic results can help us determine, are these cases related? Is there a genetic similarity between these cases? But more importantly, can we use that information to rule cases out, right? Are they in fact, potentially infected with, Campylobacter, for example, in this image, from a different source? And so were there multiple introductions of Campylobacter, into a particular community? Were there different exposures, or was it there, in fact, a common food source that exposed everyone? So it's really important when doing outbreak investigation to pair those genomic results with additional epi investigation information. So all that epi metadata, once we've integrated it, how can that help us better understand disease transmission in a specific outbreak or outbreak setting? And so again, surveillance systems can help us identify outbreaks using whole genome sequencing or next generation sequencing, or those outbreaks that we identify through epi investigation or just disease case surveillance, we can get isolates and sequence those after the fact, right? And then use, again, that additional information to change our infection control recommendations or to change our outbreak guidance to the community in which who's affected by that outbreak. All right. So this is just one example of a foodborne outbreak. This was an E. coli outbreak back in 2018. And so you can see the epi curve here in terms of the number of cases that were involved. This was 62 cases across the US, and this was an E. coli 0157H7 outbreak. The cases were all linked via whole genome sequencing. The exposure data was collected about all these individuals. About 83% had eaten romaine lettuce, and there was a trace back that had indicated that the lettuce was grown in Central California. So you can see the state breakdown here in terms of all 62 cases, what their state of residence was. But why I'm giving this one as a specific example is because they were also successful in isolating E. coli from an environmental isolate. From the agricultural water that was used as irrigation water for that lettuce. And so this is a really interesting outbreak. It's pretty old at this point, but based on the information that I've given you so far about this specific example, could you look at this phylogenetic tree and this SNP matrix together, right? We'll talk about later why that's really important. You don't want to look at them one in isolation, either the SNP matrix or just the phylogenetic tree. We want to use both in tandem to help us interpret these genomic results. But could you tell, based on what I've told you, which isolates are part of the outbreak? This is just a teaser for the later lessons. I find this, there's much information lacking here in terms of my EpiMind, in terms of understanding and being able to look at all of these identifying numbers here. I can't really pull a lot away, right? So I would immediately want to start decorating this tree with, patient ID numbers that mean something to me based on my surveillance system. I would want some, location or exposures on this. If we could, I would want a lot more information that would help me sort of pull away. You can look at the SNP matrix and you can look at those that are less than 10 in terms of the SNP differences and try to correlate the labels over onto the left and looking at the phylogenetic tree, but it's not easy to look at this and really immediately be able to apply or interpret these results. So the point here is just to say that, we need to have that epi context. We need to have a common language, as we're speaking about genomic results, as we start to apply them, whether that be for ongoing surveillance or for cluster detection and outbreak response. So in summary, it is important to understand, again, the reason for testing. Why are we doing this? What are the public health questions that we're asking and that we're trying to interrogate before we start, looking at these genomic results and applying them and interpreting them along with our epi metadata? And as I said, our two main focuses from these lessons have been understanding how sequencing can be applied to outbreak investigation, as well as cluster detection and response. But they can definitely assist us with understanding how cases are linked and whether they're genetically similar Many different surveillance systems already use genomic results, and sequencing can also assist us with food source attribution as well as confirming exposure sources. Lastly, what's most important is that we learn how to integrate this data a little bit more efficiently, whether that be electronically or automated or not. A lot of genomic data integration is still manual in public health, which is okay, but trying to look at the genomic results in isolation or just our epimetadata, we can go a lot further if we're able to integrate all that information together and make better conclusions. So that concludes our first lesson, applications in public health, and we hope that you will join us for the next 3 lessons in this series. Thank you.